The hypothalamus is the control center for autonomic and endocrine homeostatic systems such as weight management, emotional response, energy expenditure, and hormone secretion. There are 5 areas in the hypothalamus that play a role in eating behaviors and energy metabolism: arcuate nucleus, paraventricular nucleus, ventromedial hypothalamic nucleus, dorsomedial hypothalamic nucleus, and lateral hypothalamic area. Neurotransmitters that turn on feeding are called orexigenic and include neuropeptide Y and a Gaudy gene-related protein. NPY is the most abundant neuropeptide in humans, is mainly produced in the ARC and stimulates intake of food and reduces energy expenditure by inhibiting sympathetic nervous system activity in brown adipose tissue. As we consume food, both neurotransmitter levels fall, as does the desire to eat. Melanin concentrating hormone is produced in the ARC and LHA and binds to melanocortin receptor subtype 4. Genetic defects in this receptor can cause up to 6% morbid obesities. Orexins, also produced in the LHA, stimulate food intake and regulate sleep. Ghrelin synthesis is seen to increase at puberty, corresponding to the increase in fat consumption and body weight. Like NPY, ghrelin secretion increases during times of food restriction. Dynorphin, also known as the opioid peptide, promotes eating and has pain-reducing properties and acts on feeding centers to stimulate the consumption of highly palatable foods such as fats. It may also play a role in stress-induced eating. Dopamine is the central chemical in the brain that regulates how we perceive and experience pleasure. During pleasurable moments, it is released, causing a person to seek out desirable activity over and over again, like eating, especially foods high in fat and sugar. Norepinephrine acts on the PVN to stimulate eating and is released by sympathetic nervous system fibers throughout the body. Studies show overeating increases the release of norepinephrine in the brown adipose tissue, which causes diet-induced thermogenesis. The opposite occurs during food restriction, resulting in reduced energy expenditure and possibly weight gain. Ghrelin, secreted mainly in the stomach, reaches peak levels just before eating. As soon as it begins to empty into the duodenum, ghrelin levels drop. Ghrelin activates the limbic, pleasure and reward system of the brain. Ghrelin levels increase after fasting or caloric restriction and decrease following gastric bypass surgery. Neurotransmitters that turn off feeding are anorexigenic and include pro-opiomelanocortin, regulates the amount of nutrients and calories taken in, and when activated can prevent food intake. Many neurotransmitters are secreted by POMC neurons and can be activated by leptin and serotonin and can cause a reduction in food intake and induce satiety. A melanocyte-stimulating hormone is produced by the pituitary gland and can suppress appetite by acting on receptors in the hypothalamus. This effect is enhanced by leptin, a hormone released from fat cells. Cocaine and amphetamine-regulated transcript is regulated by several peripheral peptide hormones involved in appetite regulation, including leptin, cholecystokinin, and ghrelin. CART and cholecystokinin have synergistic effects on appetite regulating, and gene mutations are linked to eating disorders and include obesity and anorexia. Serotonin. Tryptophan is required for the production of serotonin. Serotonin is synthesized in neurons of the central nervous system where it regulates mood, appetite, and sleep. Cholecystokinin is secreted by the duodenum where, when fat is present in the digestive tract, it stimulates fat-digesting enzymes. It also inhibits neuropeptide Y secretion to slow eating. Peptide YY is secreted farther along the intestine after eating and inhibits neuropeptide Y secretion similarly to CCK. Bombacin, secreted by the stomach, glucagon and amylin, secreted by the pancreas, produces short-term satiation. Overactive feeding centers or increased production of certain neuropeptides could lead to excess consumption of food, binge eating, food obsession, and reduced or increased energy expenditure. A variety of studies have found alterations in serotonin system in individuals with anorexia have found that they have lower levels of serotonin metabolites in individuals without the disorder, and found that higher levels of serotonin correspond with levels of anxiety and obsessive behavior, and that they are more likely to carry a particular variant of the 5-HT2A serotonin receptor thought to increase the amount of serotonin in the brain during the non-starved state. Another article said starvation actually makes people with anorexia feel better by decreasing the serotonin in their brains. As they continue to starve themselves, the brain responds by increasing the number of serotonin receptors to be more efficient, utilize the remaining serotonin. So in order to keep feeling better, the person needs to starve themselves further, creating a vicious cycle.